Hey, my name is Zhu Yao, and I'm one of the two leaders of this year's CPU China team. Here, I'm going to introduce to you what we are trying to do. I'll start with microRNAs. The majority of microRNA genes are transcribed by RNA polymerase II in the nucleus, and the primary microRNAs are kept spliced and polyadenylated. These long primary microRNAs are cleaved by microprocessor, which comprises the double-stranded RNase 3 enzyme DORSA and its essential cofactor, the double-stranded RNA binding protein DGCR8. Microprocessor recognizes the single-stranded RNA stem junction as well as the distance from the terminal loop region. It specifically cleaves the double-stranded RNA at about 11 base pairs from the junction with the flanking single-stranded RNA to produce hairpin-shaped precursor microRNAs. The precursor microRNAs are then exported from the nucleus to the cytoplasm by exportin 5 and further processed by dicer 1 into the mature 22 nucleotide microRNA duplex. After the assembly with the Argonaut proteins 1 to 4 of the microRNA-induced silencing complex, one strand of the mature microRNA is bound by an Argonaut protein and retained in the microRISC to guide the complex to complementarily target messenger RNAs. Argonauts are localized to specific regions in the cytoplasm called P-bodies, which are regions with high rates of messenger RNA decay and are where post-transcriptional gene silencing actually occurs. The pathway that I have just showed is also called the RNA interference pathway. Multiple researchers have been attempting to exploit the therapeutic applications of RNA interference, and specifically looking to target genes that cause various types of cancer. Using this mechanism, researchers can cause a drastic decrease in the expression of a targeted gene. However, RNA interference itself lacks the specificity for discriminating normal cells and cancer cells. Well, basically, you want to kill the tumor cells, but you end up killing everything, which can lead to severe side effects. Besides, constitutive knockdown of many genes may also result in undesirable outcomes, especially in biomedical applications such as gene therapy. Therefore, spatial and temporal regulation of RNA interference are of significant importance for basic research as well as practical applications of RNA interference. We, CPU China, this year has come up with a scheme to do that. The scheme involves a RNA-dependent RNA polymerase. Well, what you probably heard of the most is the DNA polymerases. These enzymes are essential for DNA replication and can create two identical DNA strands from a single original DNA molecule. But there are all RNAs containing viruses with absolutely no DNA stage. What they need for replication, though, is the RNA-dependent RNA polymerases. In such virus as hepatitis C virus, the non-structural protein 5b plays the role as an RNA polymerase. NS5B could not direct de novo RNA synthesis without an RDRP-specific RNA promoter, which contains a stable secondary structure and a single-stranded sequence that has at least one 3-citidylate. Hepatocellular carcinoma is the fourth most common cancer and ranks second in cancer-related deaths globally, considering malignancies, severity, treatment challenges, and 5-year survival rate. Like all cancers, HCC is mediated by somatic aberration in the host genome. Since gene therapy is a type of treatment which uses genes to directly correct these aberrations to treat illnesses, it is not difficult to explain the enthusiasm about gene therapy among cancer researchers. How we treat HCC in a conditional way is that we express primary microRNA analog in the cancer cells, but in the meantime we block the basal segments by treating the primary microRNA with an oligonucleotide that is complementary to these segments. In this case, dorsal processing of the primary microRNA is blocked, so no RNA interference effect will be observed. If the NS5B RNA polymerase appears, though, with the ability to direct the novel RNA synthesis, the primary microRNA is relieved to be processed into precursor microRNA and mature microRNA, hence the interference effect. Here we choose MAP4K4 as our specific molecular target, which I'll cover later. How we achieve that specifically in cancer cells is that we use cancer-specific promoters to guide the expression of the aforementioned parts. We use one for the expression of NS5B, another for the primary microRNA analog and inhibitory strand, so that only HCC cells will be the target of our conditional RNA interference. This is called the AND gate. MAP4K4 is quite a novel therapeutic target in cancer. It plays an important role in two-thirds of all the different tumors. Knockdown of MAP4K4 proves to affect the expression, activity, or function of many factors that could act as a downstream effector or signaling mediator. But generally, inhibition of this kinase limits the cancer's malignancy. It should be noted that neither the molecular target nor the specific promoters are essence of our scheme. Well, you can always try different targets and disease-specific promoters to apply the scheme to other types of cancer, Parkinson's disease, atherosclerosis, and so on. Indeed, such significance has prompted us to sign for the new application track in this year's competition, rather than the therapeutics track one would assume